Okay. Are you presenting, Dimitris?
It's a team effort, but
Yeah,
yeah,
first.
I'm sharing the screen.
Fantastic.
Oh, can you see the screen?
Yeah, perfect.
Yeah.
Okay.
So yeah.
Okay. Hi everyone. Okay. Hi everyone. I'm going to present the work conducted for the hands-on one, cartilage multiscale modeling from human body motion to tissue turnover. So first, a little bit of introduction. Could you? Okay, thank you. Articular cartilage is the highly specialized connective tissue of the arthrodial joints. Its principal function is to provide a smooth, lubricated surface for articulation and to facilitate the transmission of loads with a low frictional coefficient. It is devoid of blood vessels, lymphatics, and nerves, and is subject to a harsh biomechanical environment. It is composed of a dense extracellular matrix, which consists of water, collagen type 2, and proteoglycans, with other proteins present in lesser amounts. These components help to retain water within the extracellular matrix, which is critical to maintain its unique mechanical properties. Therefore, an accurate estimation of cartilage mechanics is important when analyzing the function of the knee joint and when studying the risks for the onset and development of the disease due to the cartilage damage. Thereby, in this hands-on,
Oh, sorry.
Thereby, in this hands-on motion analysis has been done in the UPF MOCAT labs, and knee joint forces have been calculated from inverse dynamic analysis. These forces have been further translated as boundary conditions on a cartilage tissue finite element model. Multiphysics simulations performed with VPHi had allowed us to introduce the effect of pre-existing tissue damage, for example, the osteoarthritis, which is characterized by the progressive degeneration of articular cartilage. Then the calculated mechanical fields have been used to fit an agent-based model of cartilaginous cells developed in NETLOGO, so as to explore the catabolic response of these cells in terms of inflammation and protein expressions. Now my classmate is going to explain a little bit more in deep the first part.
Okay, so the first part, as mentioned before, was focused on the study of the whole body while generating a gait cycle. To do so, many sensors were attached to the volunteer's body to be able to keep track on its movement, while walking and limping over a force plate to capture the motion and obtain different ground forces. Normally, we would have done this, and one of us would have been the volunteer, but due to COVID situation, the files were directly given to us. Then we opened the files on a Smart Clinic to create a computational model where we could watch videos of the sensors imitating the volunteer's motion to identify the first and the second heel strike for both right and left foot, and also the toe off with the respective times. It is important to highlight that we use only 3 of 9 walking to develop this study because this one represents a person walking, while the other ones were either limping or not entirely correct. No, go back, please.
Awesome.
Don't worry. This methodology would have been exactly the same for the limping, but due to some time constraints, the data were given to us. Next, we use a smart analyzer to obtain certain data tables, such as ground reaction forces, especially on the left and the right knee, time per step, and mass. Finally, with this information, we went ahead to calculate some important data in order to proceed to part 2, tissue analysis. Okay, these results show the gait of the volunteer when walking, limping, and running. As it can be seen, the forces are normalized considering the mass of the volunteer, and the x-axis represents the number of samples. It should be noted that the time is larger when the volunteer is limping due to their difficulty to complete the gait cycle. On the other hand, the force is higher when the volunteer is running since the load on the knee is 3 times bigger than the walking, and the lowest value were obtained in the limping scenario with almost no noticeable difference between the maximum and the minimum peak. With the aim of sorry, can you change the slide? Okay, thank you. For proceeding to the tissue analysis, some data was calculated. So first of all, with the peak were taken into account since they represent critical points, and the plateau was also analyzed. In this slide, we can appreciate the pressure generated in the medial and the lateral cartilage for each different gait cycle. to calculate the pressure where the force was denormalized using the mass of the volunteer and the area of the cartilage was considered. Each cartilage is supposed to be received 10.25% of the force that the knee supports. And as expected, the running gait has the largest values.
Continuing to the second part of the hands-on, we proceeded to employ a finite element analysis to find out what happens in a tissue level. And for that, we employed 3 different simulations for normal, gait, limbing, and running, with the data coming from the previous step. So we simply translated forces into stresses by dividing with the area, and simplified to linear regimes for the sake of computational time. So were we to present the boundary conditions, that they would be fixicity on the bottom of both the medial and lateral cartilages, and the stresses are applied on the top. And both of the cartilages are saturated. So to do that, regarding the constitutive law, we employed the simplest biphasic model we could do, and that would be a neo-Hookean combined with fibers that are in a random 45 to minus 45 degrees directions, and the effect of the chemical potential and the osmotic pressure due to the saturation of the cartilages. And the results, yeah, they're animated. And the results regarding normal gait, running, and limbing can be seen here. And these are pretty much the effective fluid pressures inside the cartilages. And we can see that regarding stresses, regarding the effective fluid pressure, at least for the normal gait and the limbing, the results are pretty much the same, while for running, the stresses are way, way higher. And if we were to pretty much introduce a probe that goes inside the cartilage and measure the stresses along the cartilage, then we could see this sandwich looking concentration of stresses. So the stresses are low, close to the boundaries, then they increase, decrease in the middle, and then repeat the same pattern to the other side. And essentially, we used these values of the probe, let's say, for the agent-based model. So the input of the agent-based model would come from each one of those graphs, where we can see that, for example, the peak, let's say, fluid pressure is 0.3 for running and close to 0.1 for gait and limbing.
Okay, so in the last part of our hands-on, we will jump from this tissue level that Dimitrios was talking about to the cellular level. For that purpose, we firstly describe our network as a single elements divided in different submodels, including the mechanical load, cell death, glucose metabolism, stoichiometric metabolism, and cell stimulation that interact between them, activating or inhibiting. So once this network is defined, the next step was to use a mathematical approach that was from Mendoza-Xenarios in order to jump from these single elements to a continuous dynamic system. And in the end, we used the agent-based modeling in order to represent this continuous dynamic system, and we use this NETLOGO software for that purpose. So according to the inputs parameters, we established that our model will start with 4.2 million of chondrocytes per cubic millimeter, and we told to our program that we want that the experiment should take like 4 hours. In terms of metabolic conditions, we defined for walking, running, the following conditions that we can see in the first table. So we include 5 micromolar of glucose concentrations, 5 kilopascal of oxygen tension, and a pH of 7.1. In contrast, we expected lower values for the limping conditions. And finally, in terms of mechanical loading, these data were obtained from the second part of the hands-on that Dimitrios just mentioned. So according to the results, we can see the data normalized to the walking condition that we assume that it's, or we said that is 100%. So first of all, we can see a downregulation of aggrecan and collagen 2 in the running condition, but we can see this downregulation even stronger in the limping condition. On the other hand, we can see an upregulation of collagen 1, MMP, and ADMTS in running condition, and this upregulation is also even higher in the limping case. So what we can say about these results, that we can see a tendency towards a catabolic phenotype where the extracellular matrix producer genes are downregulated, and the genes responsible for the matrix destructions, such as ADAMTS4 or MMP, are increased more in the limping condition than in a running. So in the end, I will, no, next slide, please.
Yes,
Thanks. So I would like to mention finally some limitations that we found in our agent-based modeling. First of all, I would like to say that our metabolic condition inputs for limping case are based on our assumptions, knowing empirical data. So we can say that this experiment was kind of an approach. Then in terms of our software, it was not possible to include the values that Dimitrios showed, because it was not possible to write the whole value with all the decimals. So we were kind of forced to round these values. And in the end, I will also mention that our agent-based modeling is based in two-dimensional space. So of course that it could, we are using this as an approach, but it will not behave as a three-dimensional environment that it's like the physiological one, I will say.
So therefore, in this hands-on, the organ, tissue, and cellular level has been employed and integrated with the aim of analyzing and obtaining an holistic representation of the osteoarthritis progression mechanisms. Multiscale modeling has allowed to tackle osteoarthritis from different levels at the same time, to have a better view of the pathology, since the different levels are interrelated and do not act as an independent unit. It has been seen that the forces and time obtained from the gait analysis at the organ level can be utilized as a boundary conditions of the finite element model at the tissue level. Finally, the outcomes as the pressure, frequency, etc. were used for the agent-based model, allowing to observe the conditions at the cellular level, as the concentration of collagen, aggregants, etc., which are main components of the extracellular matrix. Even though there are limitations and assumptions with this workflow, we are able to develop a more patient-specific treatment approach according to the conditions of a certain patient. This way, the patient's quality of life can be improved. And in terms of clinical trials and experiments, the financial impact can be reduced, as well as the employment of animal models. And this will be all. If you have any question, we will be glad to answer it.
Okay, so thank you very much. Unfortunately, we will not have time for questions. So I think we will move then directly to the presentation of hands-on 2. And yeah, congratulations on the work and on the nice presentation.
Thank you.
Thank you.
So the hands-on 2 team, are you guys ready? Yeah.
Yes. Can I share the screen?
Yeah, fantastic.
Just a second. Can you see it?
Not now.
Not yet. No.
It's loading. Yeah,
Okay, perfect.
yeah, it works. Yeah.
So, hello everyone. My name is Maria Cristina, and today, together with my colleagues Sofia and Kirsten, we will present you the hands-on entitled Thrombus Formation in Left Atrial Appendage in Atrial Fibrillation Patients. To start with, we would like to emphasize on how this hands-on has been a really great opportunity for us on many aspects, such as working together as an international team or learning useful information for our academical work. Perhaps one of our questions would be, do you have any other hands-on for us? So thank you for your implication and for offering us this opportunity. Coming back to medical aspects, when we have a healthy heart beating, it means that it beats regularly under the control of the electrical impulses. These impulses radiate from the top to the bottom of the heart in a coordinated way. In the case of atrial fibrillation, the electrical signals are abnormal, radiating through the atrial walls in an uncoordinated manner. This causes the walls of the atria to fibrillate rather than contract normally. And in conclusion, it leads to decreased flow velocities. Because of this, blood begins to pool in the atrial appendage. The pooling can create clots to form, which, if dislodged, can travel to the bloodstream towards the brain. Once they get there, an ischemic stroke may appear. Regarding the morphology of the left atrial appendage, which is the most common site for thrombus formation, it is currently classified in 4 big categories, meaning chicken wing, cactus, windsock, and cauliflower. Out of these 4, the chicken wing is the most common and carries lower risk. The aim of this hands-on was to replicate a modeling workflow in the left atrium and to find the areas most susceptible to thrombus formation. For this, we began with segmenting the left atrial by using the 3D slicer program. This is an important step because if we obtain a wrong geometry, the simulation would not offer us the precise information we'd be expecting. We've been performing the segmentation both manually by painting with a sphere brush and in a semi-automatic way with the threshold option. We have also been using a couple of more tools in order to improve our segmentation. Once we had our geometry ready, we continued with generating the mesh. And now I will let my colleague Sofia explain this part.
Yes, so the next step in our workflow was creating the mesh. And we created the mesh using tetrahedral elements, which are, so when you're creating a mesh for the CFD simulations, you can either use tetrahedrons or you use the structured mesh, but since the geometry of the left atrium is very irregular, it's better to choose tetrahedral elements. And before we could create the mesh, we first needed to improve the geometry a bit so that it's easier for the solver to solve. And one thing that we did was that we cut the pulmonary veins and the mitral valve so that we had flat surfaces with sharp edges so that the solver later knows where the flow is and where the walls are. And to do this, we used the programs called Mesh Mixer and Mesh Lab. After we had created a surface mesh in those 2 programs, we used the software called GMesh to create the volumetric mesh. Since we were using the ANSI student version for our CFD simulations, we were limited to the number of elements. And therefore, we couldn't do a so-called convergence analysis where you would create several meshes with a different amount of elements to find what resolution you need to get accurate results. So we just used the maximum amount of elements possible. And now I will go to explain the Fluent Simulation. So can you go to the next slide? Yes, thank you. So in the next step, we used the mesh that we created and opened it in ANSYS Fluent to do the CFD simulations. We simulated the blood flow as a transient simulation, which means that we simulated it over time. And we assumed that the blood flow in the left atrium was laminar flow, with blood as an Newtonian fluid and a constant density. The time step of our simulation was 0.01 seconds, and since the length of the cardiac cycle was 0.85 seconds, we had 85 time steps in our simulations. An important aspect in CFD simulations is how you define your boundary conditions. And they were, and we chose to have a pressure boundary conditions at the pulmonary veins, which you can see in the blue curve on the screen. This is a general boundary condition and not patient specific. At the mitral valve, we prescribed the blood velocity over time, which you can see in the red diagram. Additionally, we also prescribed the motion of the mitral valve based on the yellow curve. Yeah, it should be noted that there is no consensus on how you define the boundary conditions in the left atrium. So it's also in other publications was used that you prescribe the flow in the pulmonary veins instead. So we tried to do these simulations on our laptops at home, but since these simulations are quite demanding, it wasn't possible to do the simulations for all of us. And we also only simulated the first cardiac cycle, which meant that we more had like an initial state. So for doing our later flow analysis, our supervisors provided us with results from simulations where they ran 3 cardiac cycles so that we could analyze the third cycle, which is considered to be more accurate. Yeah.
So I will hand over here, take over. Okay, so for post-processing of our simulation data, we used an open source software called PAView. And this is an application very powerful for analysis and visualization of large data sets. And we had several possibilities to visualize our simulated flow inside our model. So here, for example, you see the velocity vectors that are represented with respect to the magnitudes and the vector fields as a volume as well. Other possibilities would have been, for example, to visualize streamlines, which are curves that at every point are tangent to the respective vector field. So basically they represent the path of a particle going through this vector field. And it's also possible to simulate actual particles that are subjected to the vector fields and thereby create these path lines. But we chose this velocity visualization for this presentation. So that's what you see here. And we got different data sets. So here we compare 2 different cases. So you can acknowledge different courses of the simulation. But of course, if you would switch to the next slide, we wanted to explore our CFD simulation in regard to our initial question, if it's possible to predict sites that are more prone to thrombus formation. And for this purpose, we looked at the endothelial cell activation potential, or short ECAP, which is a numerical indicator for thrombus formation. More precisely, it's defined as the ratio between the oscillatory shear index, which represents changes in the wall shear stress field direction due to complex flow patterns, and the time average wall shear stress magnitude, which is directly connected to the velocity. So ECAP can be interpreted in the way that higher ECAP values correspond to an increased endothelial susceptibility to thrombus formation. So with that in mind, when we have a look at our 3 data sets, we would definitely expect thrombus formation in case 6, as you see that in the area of the appendage, there are definitely increased values of ECAP. And while in case 3, it only has small spots with a higher ECAP, and case 1 seems not at risk at all. So in conclusion, thank you. We have learned a whole lot this week. So from segmenting 3D image data to obtain the desired volumetric model, and then processing this volume to a high quality mesh, and yeah, learning how to set up the CFD simulation with our mesh in ANSYS, and subsequently also to visualize the output to allow conclusive evaluation. And yeah, it was a lot of input, and in the name of our whole group, I want to thank Andy, Mireia, and Xavier for their efforts to show us all of this and their patience with our questions and yeah, technical issues at times. But we know that yeah, it can't have been easy to arrange this virtual hands-on. So yeah, I'm still positive that we made the best of it. And yeah, I wanted to thank you again. And of course, also the organizers of this lovely summer school that made this all possible from the beginning. And this would be the end of our talk. So I don't know if we have time for questions or if this maybe happens later on.
Okay, great. Thank you very much, guys. Very nice work. Congratulations. So do we have a hands-on 3 team ready?
Yes.
Okay.
I can share my screen. Yes. Okay. So hello, everyone, and thank you for having us. We are the group 3, Katarzyna, Monica, and Marta. And our hands-on was focusing on robotic arm programming and tissue modeling for image-guided brain surgery. And our instructors were Jerome and Mario. So the specific problem that we focused on is robot-assisted stereoencephalography, or SEEG, which is an invasive monitoring methodology in which typically over 10 intracerebral electrodes are implanted deep into the brains of patients in order to precisely record electrical activity in the brain. This method is typically used for pinpointing the specific epileptogenic zones in the brains of patients who have a focal epilepsy but do not respond to classical treatment options. So that is why this invasive treatment or invasive method is necessary. In this case, surgical resection of the epileptogenic zone is the only way to help the patients, and SEEG is beneficial as a tool for pinpointing the precise brain area to target with this surgery. Robot-assisted SEEG uses a robot that places a guide for the drill precisely in the targeted spot of the patient's skull, and the neurosurgeon then drills holes for the electrodes through this guide. This technique is more precise and as safe as classical SEEG without the use of robots, but it presents an added benefit of a significant reduction in time that is needed for this procedure. One of the challenges of this technique that is left to tackle is the trajectory planning and the displacement of electrodes post-implantation due to edema formation, or that is swelling of the brain, which happens in the location of the injury that is caused by the electrode, as this technique is quite invasive. So all in all, the workflow of this technique is as follows. Firstly, a volumetric and high-resolution MRI scans are obtained before surgery of the patient and are transferred to the robot's software. Then a simulation of guide placement within the pre-selected zones of interest in the brain and electrode displacement and brain swelling is ran. Secondly, the selected trajectories of electrode implantations are uploaded to the robot. And lastly, during the procedure, handheld drills are introduced to the skull through the robot's guide and electrodes are implanted. My colleagues will now tell you more about what exactly we did during our hands-on. Yeah. So this project has involved different aspects, ranging from image processing, robotics and programming, to finite electron modeling, so biomechanics. In particular, regarding the robotic planning,
to reach the aim to reproduce virtually the placement of the electrodes in the skull with the use of a robot. We installed different tools. One was Unity. So this is a game engine, and so we can simulate a virtual environment. Here you can see the patient and the robot. Then there is the robot operative system and called ROS, and ROS and Unity has to communicate, so to exchange information, and this has been reached through the use of the Docker. Then also the images, the data are needed, so to be able to import these images and the position of the reference for where to put the electrodes, and this has been done through a Python script using some libraries such as VPHi, and also a communication so as to be established between the script, the code, and the ROS, so we can control the robot in this way. So briefly, from the Python script, then we add the position of the electrodes, and then we have to communicate to the ROS and to the Unity to start the simulation. What we have done was here, as you can see, an example of the communication between ROS and Unity. So with the creation of a button in the Unity graphical user interface, and then this button is connected to some functions in ROS. So we were able, clicking to this button, to print the pose of the robot, and similarly also to program the robot so it can follow a trajectory from an assigned position to another. There was no time to reach the final aim, that was to import the skull and simulate that the robot can grab the electrode and put it in the reference position that we talk about in the skull. So here you can see some previous works results, but what we have learned is to connect all these tools together. And then we can also, from previous works and in vivo experiments, these procedures can reach high accuracy. And these virtual environment settings, let's say, we think that can help to improve the accuracy of the planning, so in such a complex procedure. Also, but some improvements can be done, such as put the skull in the correct position, like where the patient is, but it involves other steps like registration, so there was no time, but then we focus on another aspects of finite element, and Marta will talk about that.
Yes, thank you. In the second part of our project, we tackled a surgical complication, which is sometimes happens in the patient after the surgery, which is a swelling-induced electrode shift. So basically what happens is that due to the insertion of a drill in the patient's brain, there is the development of pressure that is due to the liquid accumulation around the area that has been drilled. And this can affect the pre-planning, because the preoperative Preoperatively, the target is positioned looking at images where the brain is in its normal condition, so there is no brain shift. But we know that after 72 hours, we have a shift in the electrode position due to this swelling phenomenon. So we wanted to find a way to predict the electrode shift and possibly to propose a way to avoid this problem, or to estimate if this problem can really affect the surgery. So next slide, please. Thank you. So to tackle this problem, we developed a finite element model, so a structural model. We basically idealized the geometry of the brain as a half a sphere. And we defined 4 layers, which correspond to 4 different structures of the brain. So we have the innermost layer, which is the basal ganglia, the corpus callosum, the corona radiata, and the cortex. All these layers have been modeled with a biphasic constitutive law to take into account the fact that brain is a porous material, and so it has a poroelastic behavior. So we have modeled these materials as hyperelastic and viscoelastic. And to do that, we use the PhiBio property. So PhiBio is a software for open source software for finite element modeling. And it allows to superimpose different constitutive laws to obtain, let's say, personalized material properties. And in addition, the other phase of to simulate the other phase of the material, so the fluid phase, we added poroelastic behavior. So we defined a permeability coefficient. And for this step, we have considered the material constant that we have found in literature, and we have converted to the PhiBio standard properties. No, can you go back? Thank you. So we also defined the boundary conditions, of course, for our model. And we have assumed that there is no outflow or inflow of fluid around the surface of the brain. So we have assumed impermeability on the outer shell. And we have also fixed the displacement of the whole outer surface in order not to have our brain to move around in the space. And we loaded our brain with a swelling pressure that we defined in a specific area, which is basically representing the surface around the point where the electrode is inserted, and a line that follows its trajectory inside the brain. And this pressure was about 19 millimeters of mercury. So next slide, please. Here you can see the results. So on the right, you have our simulation result, where you can see that after 72 hours, the swelling pressure has caused pressure around the electrode position, and this has caused a shift in the tip of the electrode, which we haven't simulated, but we can see that in the point of maximum depth, the brain suffers from a 0.65 millimeters shift. And this is in the same range as values that we have found in literature, where they evaluated in different patients what was the electrode tip displacement. And so, yes, we can go to the next slide. After this workshop, we have learned how to connect a simulated operative room with a robot operating system through the software's Unity and the Docker containers. We also have looked into the fundamentals of trajectory planning with VPHi. And we have seen a method to simulate a possible planning complication due to brain swelling. And to do that, we have also learned what are the mechanical properties and constitutive equations of the brain tissue. We have achieved some results, such as we have connected the virtual robotic arm to the ROS, and we were able to give instructions to it. We also virtually placed the electrodes in a patient's skull model. So we did some pre-planning. And then we built from scratch a FEM model with the literature-based data. So that involves also a little bit of literature research. And finally, we ran a simulation of electrode-induced brain edema and quantified the electrode shift after 72 hours. So that was it. I think this week was very hard for many of us, but we have managed to learn a lot of new things. So I would like to thank our instructor and all the organizers of the workshops. Thank you.
Okay, thank you very much. So very nice. And congratulations on the work done and during this last 15 hours. It has been intensive. So do we have the team for hands-on 4? So hands-on 4, we have 2 presentations. I don't know who will be starting. Osteoarthritis, atherosclerosis.
Right. Maybe team 1, I don't know.
Go ahead.
We may, how you call it, if they are ready.
Are we team 1 then? How about our group, atherosclerosis?
Okay, good.
Good morning. This will be the presentation about multiscale in silico models of early atherosclerosis. This will be firstly our pipeline that we are going to follow. And we will start with the clinical problem and first some background. Atherosclerosis is a chronic disease that affects the arteries, characterized by inflammation and retention of lipids, forming atheroma. This atheroma contains low-density lipoprotein, macrophages, smooth muscle cells, platelets, and debris. This plaque can cause arteries to narrow, blocking blood flow, and this can lead to serious problems, including heart attack and stroke. The process of plaque development begins with a lesion in the endothelial layer that allows LDL to move into the intima and become oxidized LDL. After entering the intima, monocytes differentiate into macrophages, which have an affinity for the OX-LDL. The ingestion of large amounts of OX-LDL transforms the fatty macrophages into foam cells. At the same time, high-density lipoprotein also enters the intima and becomes oxidized. HDL helps prevent atherosclerosis by removing cholesterol from foam cells, limiting inflammatory processes in atherosclerosis. It is relevant to specify the risk associated with combined levels of LDL and HDL. And this is what this project addresses, which is modeling the 3 initial years of development of atheroma and atherosclerosis using a combination of network and agent-based modeling.
So now we'll explain briefly what these 2 types of modeling are. So network modeling is basically a way to define the relationships that happen between the items of your system. In our case, these items we have are the molecules that are involved in the atherosclerosis process. And agent-based modeling is a way to define cell behavior, which is, it's basically the way we are going to make cells decide regarding the environment they're in. So in the next slide, we can see the network that we are defining. In the network, we can see the agents of our network, which are the cells, and we can see them in black squares. We can also see these items, the molecules, which are in blue squares, and we can also see the interactions or relationships between these molecules. So the green arrows basically mean a positive interaction or an activation, meaning that this molecule is activated by the presence of this other molecule, and red arrows mean the opposite, a negative interaction or an inhibitory one. So if we go to the next slide, we can see the equations that model these type of networks, which are the Mendoza-Xenarios equations. They look like the one you see on the top right. We're not going to get into much detail, just so that you know that these equations model the quantity of a molecule regarding the quantities of molecules it has around them, more or less. And it's this interaction is modeled via the alphas and betas that you can see in the lower equation, that is then carried to the upper equation. And these alphas and betas are actually the how we quantify these interactions. You can see the table we've used to quantify these interactions. Green squares, which are the alphas, represent the activations, like the green arrows we saw before, and red squares represent the inhibitory effect, which are the red arrows we saw before. So this is all about the network. And now if we talk about agent modeling, we did this by using Boolean rules. That is, we had to define the behavior of the cells. And to do so, we actually programmed what a cell should decide once it checked its environment. So we are showing just one example, the macrophage. So just I'll explain it briefly, what a macrophage has to do. So a macrophage basically has to check if it has some oxidized LDL around it. If it does, it engulfs it or eats it, which is the same. And if it eats too much, it reaches a certain threshold and it becomes a foam cell, which and the accumulation of foam cells is what becomes the plaque. If it doesn't reach this threshold and it keeps going and living, it reaches its lifespan and it dies and it bursts and it allows all the oxidized LDL it had inside it out. So this is just one of the rules we had to program for our cells to do.
Okay, so now into the in silico technology. To be able to simulate this system, we develop our code in the open source NETLOGO programming language, which is useful for agent-based modeling environments. It has a visual, nice interface where you can explore sliders, graphs, dialog boxes, and more. We are using the 3D NETLOGO, and in here you can see an example of the interface we developed. In the 3D view, we can see the endothelial cells layers in yellow and the small muscle cells layer in red in the bottom. And the black space between them would represent the intimal space in the vessel. If we zoom in, we can see the LDL and the HDL molecules, which are in red and blue triangles, respectively. And in the slide, there is a legend for the rest of the cell agents that should appear later on the environment. Okay, so once we had the code, we thought about different trials with different values of LDL and HDL to test its behavior. The range of HDL is between 25 and 75 milligrams per deciliter and is considered healthy above 40. And in the case of LDL, it is between 95 and 200 and considered healthy below 100. So we thought that it would be an idea to just consider a first healthy case and then vary different values of HDL, keeping like an extreme value of LDL. And well, when developing our model, we saw that although the model represented was capable of predicting atheroma formation in early stages of atherosclerosis, when the conditions were favorable, we wanted to note some limitation that it has. First would be that the total amount of cholesterol also includes triglycerides, which we are not considering in this model. The second would be that we are considering multiple time scales, meaning that we are trying to span chemical reaction that happen in milliseconds in the simulation, which is 3 years long. But this is like a trade-off for computational cost. And finally, we are considering static values of inflammation rates and amount of HDL and LDL in the simulation, which is not really realistic as they could change these values overnight.
Yeah, so now we can talk about the results we achieved. Within the limited time of the summer school, we were able to create the code for our model, including the equations for all of the molecules and the rules for all of the agents in the figure that you saw before. And we implemented this code in NETLOGO environment. We were able to see the smooth muscle cell and endothelial cell layers, as also mentioned by Raquel. However, when we ran the code, we unfortunately realized that there was something wrong. There was a bug, which I'm sure we could have fixed if we had just one more day. But for the sake of time, we used Marina's NETLOGO code to simulate the 4 cases that was described before and to understand the effect of having different LDL, HDL levels in the development of this disease. So you see, for example, here the total amount of smooth muscles and endothelial cells that we have, and one single monocyte, which you saw moving around in the simulation. But yeah, as I said, this wasn't enough, so we used Marina's code, and in the proper code, you could actually see all the different agents. So all the monocytes, foam cells, T cells, LDL and HDL molecules accumulating over time. And to talk a little bit about the different cases we compared, with an LDL level of 95 and HDL of 50, we could see that there are no macrophages or foam cells indicating a healthy individual. So this is the case on the left. And when we set the LDL level super high, which is 200, we saw that the accumulation of ox-LDL macrophages and foam cells were really high. And similarly, we saw some accumulation when we set the LDL to 200 and HDL to 75. But interestingly, having the LDL higher in this case was actually resulted in a lower accumulation of ox-LDL, macrophages, and foam cells. So this was an interesting result from the simulations. And we actually see the same thing in the graphs. So in a healthy individual, there's no ox-LDL or any other bad accumulation, let's say. However, if you have high LDL levels for 3 years, you end up with a high amount of ox-LDL in the vessel. And this is also showing that having a higher HDL level is actually protective for the vessel, because there we have a lower amount of ox-LDL accumulating over time compared to having a lower HDL. So this was actually a really nice conclusion, or implication from having an in silico model for this, because we came to the conclusion that with the in silico model like this, we could actually suggest something for clinicians to look further into, such as having HDLs to be protective. And another take-home message we had was that with the combination of Mendoza-Xenarios equations and agent-based modeling, we could describe the early stages of a complex disease that actually spans multiple scales in terms of time and also size. So this was a nice exercise for us. And we would like to thank Marina and Keith, who were super helpful. And it was really nice to work with them. And with this, we conclude. Thank you for your attention. And we can take any questions maybe after everything ends.
Okay, so thank you very much. Nice work. And then now we moved into another hands-on about systems biology, which is osteoarthritis.
present this one, Jerome. So,
okay, Terence.
Can you hear me okay?
Yeah.
Just give me 1 second. Oops. How's the presentation? Is it okay? You can see it?
Yeah, perfectly. Yeah.
Okay, so we also looked at multiscale systems biology, but from the perspective of cartilage homeostasis in osteoarthritis under the excellent guidance of Maria. And I hope I can do justice to the hard work of all my colleagues in our workshop with this presentation. And thanks a million to all of them for their input. I learned a lot. So just briefly, I'll mention about osteoarthritis. We're in this workshop interested in essentially the metabolism of the cartilage of that tissue that degenerates in osteoarthritis, and obviously it's a very severe condition and affects millions of people worldwide. Of course, there are multiple psychosocial, environmental, biomechanical factors that contribute to osteoarthritis, such as knee osteoarthritis. But in this workshop, we focused on the biological factors that drive the pathogenesis, and this is something that there's still a lack of understanding on. So there's a need for new therapeutic targets, and simulations such as these network models can contribute to this. And I'll just mention I didn't know anything about these kind of models before doing this workshop, and I was sort of skeptical about how it can be representative of the complex biology, but I definitely found very useful insight into that. So this is a useful slide just to give an overview of what the whole process we went through, and this kind of encapsulates everything we did. So the idea was to collect information on the cell regulatory factors. So we looked at a fairly high level, so we didn't look at intracellular factors in particular. Generally, we're looking at pro-inflammatory, anti-inflammatory factors, cytokines, and some proteins within the cartilage tissue. And essentially, this is a knowledge-based or qualitative sort of input into the model. And that gets put together into a kind of a static graph of connected nodes with edges, which can be activating or inhibiting. And this kind of static graph can be resolved or sort of solved across a period of simulations across time to result in a kind of final steady state for how those different nodes become represented in the network, and gives us at stage 3 there a hypothetical regulatory network based on the input information, which then gets further enriched and updated to give this steady state. The final stage is to calibrate and validate that kind of hypothetical model against real experimental data and an independent validation set using, in this case, a genetic algorithm to optimize the whole network and optimize the topology of the network, the layout and the edges. So the first stage really involved collecting information from the existing literature. So for instance, within the literature, it's quite laborious really extracting what those relationships are between different cytokines and the different proteins involved. But after that basic initial network has been built, the second stage is to enrich that by going into more detail into the literature to find and try and extract a wider range of nodes and interactions between those nodes. So this is the enrichment stage of the process. And there are sort of selection criteria, and that really influences how complex the network becomes. So we use this tool string kind of to extract this information from curated databases, and it's essentially a text mining tool. And depending on how the threshold you have for the confidence you have in those interactions, you can include or exclude nodes, or in this case cytokines, or whatever aspect is going into your network. So based on those nodes and that information, obviously previous presentations have touched on this equation, but I'll just spend a little bit more time talking about the Mendoza and Xenarios equations here. And essentially, these are summing all that information that's coming into each node and applying it across the whole network over multiple iterations to find a steady state for that network. So on the left here in the upper part of the equation, you can see there's the alpha and beta inputs, which are representing either activating edges or inhibiting edges. And this would be applied in the case of a node that has both, or there might be a node that only has activators or a node that only has inhibitors. And of course, there could be nodes that have neither. But this summing of all those inputs is then applied across time with an activation function to kind of resolve the state of the whole network. So the input from that goes into the activation function. I'll just mention briefly that this H represents the gain of the sigmoid function. So a sigmoid function is used, it's standard, kind of reflecting the biological nature of the network, and it also is part of the normalization process, keeping the activations between 0 and 1. So that's the activation term and decay term. And essentially that applied to some network, whether it's the initial network, enriched network, or an optimized network, results in a steady state after a series of iterations, which might involve activation or inhibition of various nodes to varying degrees. So the second step after that has been done is to calibrate and validate this using actual experimental data, so collected from real chondrocytes and stimulated with various cytokines or other mediators. A genetic algorithm is used as a kind of an optimizing function based on the mean absolute difference to optimize the topology of the network, both for a training set and then an independent validation set, although we use a slightly modified version of the genetic algorithm, which is shown here. So it's just a summing of those errors, mean absolute difference. And from this, we can get basically a matrix of those errors for each individual node interacting with a particular stimulus or particular edge. And we need to then examine the level of error and examine for any overfitting. So in our case, we had an initial network, and this is the graphical representation that we started with. So this is just from the basic knowledge that we got from 2 publications that were put into kind of a data frame or an Excel spreadsheet. And then we used Cytoscape to visualize this, the red being catabolic and the blue being anabolic nodes within that network, and then the green being activating edges and the red being inhibiting edges. So based on this simple starting point, or at least it looks quite simple in the spreadsheet, the interactions, but obviously when it then it's graphed or visually represented like this, it's a very complex, so you can see that the many interactions that are involved. After the further kind of search for additional nodes or additional relationships using the string software tool and the text mining, you then see that the complexity of the model increases as more connections were discovered and added in. So this kind of data curation is really a key step. And as I mentioned previously, your thresholds here for the confidence coefficient will affect how complex and how many additional interactions you add into the model. But then interestingly, when you optimize the network, so this is against the training data using the genetic algorithm, the network is optimized, and essentially you can see that parts have been sort of paired away. There are now fewer interactions, but this might have higher fidelity to the actual experimental data. And this was one thing that was particularly interesting for me. And I remember Mendoza-Xenarios mentioned it in his talk, that you can have actually a subset of nodes and edges that are very much representative of even what was originally a much more complex network. And that was very interesting for me. And also it ultimately, in the case of osteoarthritis, we're looking at the balance between anabolic and catabolic factors, which is obviously particularly relevant to the pathoetiology of that condition. So those 3 previous networks I showed you can be reflected in these baseline steady states of what happens to that network when it runs over a period of time and reaches essentially a kind of equilibrium or steady state. So you can see the difference as we had started with the initial network, enriched it, and then optimized it. Now, unfortunately, our genetic algorithm and optimizer didn't converge, so it didn't. We didn't necessarily get to a true optimized network based on the data we had, and maybe some bug in the code that we didn't have time to figure out. But at least we could see an evolution of the network as we applied those different steps. So then just these error plots to finish on. We had the enriched network, which is before any optimization has been applied, and then the optimized network, and you can see the error decreasing there. But unfortunately, there still was quite some error even on the training data in this case. And during the validation step, again, an independent set, and there was also still quite a bit of error there. But this, yeah, there's going to be, it's important to observe at this stage in order to check for the bias and the variance, check for any overfitting that might be happening during the training stage. So it was a fantastic workshop. To conclude, it basically helped us to understand or help to describe the healthy baseline in the high-level chondrocyte regulatory network, and the genetic algorithm helped to replicate the experimental data. So through this whole process, these were the key outcomes, and that was particularly interesting to see. So I'll leave it at that point. Sorry for overrunning a little bit, but we'd like to thank you very much for the opportunity, and we really enjoyed the whole workshop. Thanks very much.
Nice, thank you very much, guys. Nice group picture, even through Zoom. Okay, so we move on then with hands-on 5, which is about patient classification.
Okay, great. Can everyone see the slides and hear me?
Yeah, Cameron, yeah, perfect.
Cool. All right, so I'll be firstly talking about the background to hands-on number 5, where our group was looking at the stratification of patients with complex phenotypes. Just trying to advance that. Okay. So the challenge that our group is looking to overcome is in population studies in clinical situations. So often in heterogeneous disorders, the symptoms are overlapping, making diagnosis quite difficult. There's no clear mechanistic understanding, and clinical guidelines are often rigid and take a one-size-fits-all approach. So with our methods, we want to identify different phenogroups within the population in order to better understand the pathophysiology and hopefully be able to implement a patient-specific approach and personalized medicine. So just to, again, re-emphasize that this clinical data is very challenging to interpret, but we aim to use dimensionality reduction to make it easier to interpret in this way. The data set that we were working with consisted of 4 main groups. So there was 2 control groups, 1 group of athletes, and then 2 patients, giving us a total population of 289. And within this data set, we had 11 different features, which were consisting of left ventricular outflow and inflow, and various different Doppler and strain measurements from echocardiography. And on the left-hand side, you can see that just to illustrate the high dimensionality of the data set.
So as a visual summary of what he was saying, we're going to use the features that we just saw and try to stratify the patients in different dimensions and see if we can relate these stratifications to different phenotypes. Yep. So to do that, we're going to use dimensionality reduction, which is a process in which the similitudes and the differences between the samples within a set are translated into a reduced number of features as well in another set, trying to maintain the maximum variability. So trying to maintain to keeping these differences and these similarities between the samples. Yep. Next, please. Yep. There's different algorithms and techniques to use this dimensional reproduction. The simple ones, which are linear, like principal component analysis, the one that we used. Also, there's no linear, and we can differ as well, having single view, which means we can just use a feature, or multi-view that combines multiple feature. Of multiple view and nonlinear, we are going to use as well multiple kernel learning, and try to compare both outcomes from both algorithms. Next, please. So PCA at the end, it's a method that tries to find a collection of orthogonal vector spaces in which the variability of the data is maximized, and try to represent the samples in these new features, maximizing the variability. Next.
In the same way as PCA, we also apply multiple kernel learning. This is a pipeline of MKL. First, we use the feature and transform it in kernel matrix. The sum of this kernel matrix create a global kernel matrix, and then we can apply the MKL algorithm, and with the projection matrix and weight, we can have an output space where after this output space, we can, next please, apply a clustering. Here the clustering used is a k-mean, and the k is the number of clusters.
Now we're on to the results. So the first step, as my colleagues mentioned, was to first look at our data using the PCA. And so we applied that to each of the 11 cardiac features that we mentioned earlier. And this is just an example of one of the features. In this case, we took regional LV strain, one of the regions of the LV, and you can see the clusters along the first principal component dimension and the second one. The different patient groups are presented in this plot. And then we explored the data space using the G input function in MATLAB. So we selected points along the horizontal axis, and you can see from the reconstructions of 2 dimensions that it's one of the main differences across the principal, across P1 is the decrease in amplitude. And if we sample data along the vertical axis, one of the key changes is the shift in amplitude along the y-axis across the cardiac, so later in the cardiac cycle. But you can also see from these peaks that it's not just as simple as shifting the peaks in principal component in P2 in the second dimension. So there's more information being encoded in the PCA dimensions than just an XY kind of axis. So to explore all the data, as you get more features and more data, it starts being quite difficult to look at every single parameter and try to understand and conceptualize it and synthesize it together coherently. So the next step was to apply machine kernel learning to the same data set. So this is the machine kernel learning output with the different clusters presented. So we trained it using the hypertensive patients, athletes, as well as the 2 healthy controls. Then we used data from the HCM patient group and projected it on the sample space, and you can on output space. You can see where the green dots are on the left-hand side of this figure, and is more overlapping with the hypertensive patients rather than the healthy controls. We can then also remove these labels and use the k-means method as we talked about, and using the silhouette coefficient to cluster the data, and 4 clusters were found to give us the greatest separation between the groups. So here are the 4 clusters in different colors, and just to bring your attention first to just to one thing, just the hypertensive patients here seem to be in cluster 1, and cluster 2 would be more of a healthy control group, and these clusters 3 and 4 in between, which will be useful in our next slide. So this is all these slides, all these figures, sorry, present the means and standard deviations of the 4 clusters for the 11 cardiac parameters that we fed into our MKL algorithm. So there's a lot of information here that you can see is being captured and encoded in smaller dimensions. And just to show you some things that might be clinically relevant or to help maybe understand patient disease progression is that there seems to be a consistent left to right shift in all our strain variables from orange, yellow, purple, and blue. And 1, 2, 3, 4, 5, 6, 7 are all strain variables, the 6 LV strain variables and the LA global strain. You can see that there's a consistent pattern. And if you remember, cluster 2 was the most healthy population, and cluster 1 was had more hypertensive patients. You can see that there's this same shift, which might be interesting in terms of looking at how disease has progressed. Another thing that these curves might be able to tell us is that there's also differences in amplitude between the different groups and might help us to better interpret individual data. So based on these clusters, we then explored some clinical characteristics which weren't used in the MKL model. And here, in this case, you can see that cluster 4 seemed to comprise more of younger participants, and this was not something that was fed as a data input when we clustered the data. But if you only had, say, the age and you looked at ejection fraction for cluster 4, it's not incredibly obvious where these patients who have lay on this in the clusterings or the disease spectrum, because there's quite a uniform distribution of ejection fraction here. So this is another example of how the MKL algorithm can help us better interpret complex data and will probably give us more information than just the standard cardiac parameters alone. So to summarize, in this 10 minutes, we've give you an overview of the concepts that we've covered in our hands-on, so principal component analysis for a single variable and multiple kernel learning to look at multiple features. We applied it to 289 patients with complex cardiac phenotypes. And looking at the data in this manner will allow us to obtain meaningful clinical insights that might not be possible if you just had a curve and did simple statistical tests like a simple t-test or only looked at peaks and time to peaks. And with that, I've come to the end of the presentation. On behalf of the team, also would like to thank our brilliant instructors, Pablo and Mariana, for all their help in getting us from not knowing anything to being able to produce this presentation and the code that we've managed to run. Thank you very much.
You're muted, Jerome.
I was on mute. Thanks. Yes, very nice. Thank you. So we move then to the last but one, hands-on 6, which is spine modeling.
Yes, Jerome, I will be presenting, please.
Okay, Alvaro.
Hi, good afternoon to everyone. Now we're going to present the work we have done in hands-on number 6 during this week. But before, we wanted to thank the organization for such interesting seminars and activities during the whole week. To begin with, this is the pipeline we're going to follow in this presentation. Lumbar lordosis can have abnormal curvatures in the spine. And currently, this condition can be fixed with a really invasive chirurgical procedure. And before moving to the next slide and explain it, I want to warn you that we're going to put a video of the surgery so that if you are very apprehensive, you may want to close your eyes or to look to another side. So focusing on the surgery, there are 2 steps. The first one is the destabilization of the lumbar spine. which consists of an osteotomy and a facet joint removal, as you can see in the first part of the video. Then in the stabilization step, the spine is realigned and stabilized using long instrumentation, like for example, screws and rods, as you can see in the video now. However, some complications can arise during surgery. On the one hand, there can be major complications, like for example, infections, and also a hardware failure when where the device can break in the osteotomy site, or there can be proximal junction failure in the upper level of the hardware. Now, knowing this risk that this procedure implies, what if, in addition, the surgeons correct the spine in a bad manner? Will the risk of mechanical failure increase? Answering this question motivates us to develop a biomechanical analysis of instrumented lumbar spine. The objective is to analyze the loads and stresses of lumbar vertebra, and instrumentation will suffer by simulating two different lumbar lordosis corrections, one of 49 and another of 69 degrees. With this mechanical assessment, we will be able to estimate the risk of mechanical failure, which can be predicted with a gap score higher than 7. Moreover, this computational tool will also be useful for getting better knowledge about the stresses after surgery, how can they affect the lumbar spinal cord, and also to study the consequences of using different materials for the used instrumentation. The main motivation for us was to assess decision-making of doctors so that they can have in silico data reference to improve surgery planning, and also to reconsider the intervention in case of mechanical failure is predicted. However, this requires patient-specific configurations, and another challenge is a process in a simulation.
So now we will go deep into the followed methodology. First of all, here we saw the general pipeline with the current techniques used to fulfill our objective. We have been working this last week in 3 of these steps, so we will go further into detail in the next slides. First, we were given the data as well as the model 3D reconstruction, and the next step was to apply a statistical shape modeling in order to specifically apply a PCA method to find the different spine morphologies. This way, we generated the 2 models we can see at the right of our screens. First of all, one spine with lumbar lordosis of 49 degrees, which is our main model, and another one of 69 degrees, used to find the kinematic constraints. Each of them had different standard deviations. After this, we applied a step of mesh morphing. First, what we did was to perform rigid registration in order to find a rotation matrix that found the relation between each of the vertebrae in the configurations of the 49 degrees and 69 degrees. Then we computed a rotation field, which served as the kinematic constraint for our model. And finally, with the thin plate splines technique, we developed the spine structural meshes. Now what we have here is the real mesh morphing for the L lumbar lordosis 49 degrees pipeline. Mesh morphing is firstly done in the vertebrae, and then it is extended towards intervertebral discs. Then finally, all parts get joined between themselves, and the whole structure mesh is obtained. So now for the firm, we use the following material properties for the annulus fibrosus and anisotropic hyperelastic material with a formulation called Holtzapfel. After this, for the nucleus pulposus, the material was poro hyperelastic, and elastic materials for the C-bip and vertebrae body was used. Finally, the ligaments were considered as hypoelastic materials, and the boundary conditions were based on an encastre and pore pressure. But lastly, but not less important, are the kinematic constraints that need to be applied in the following steps. To end up with this section, we would like to show you all the FEM pipeline. Here, the first step is the intervertebral discs for swelling, which takes quite a long time, that specifically are 8 hours. Once the 8 hours have been accomplished, the structure is in equilibrium. After that, we need to apply the kinematic constraints, as well as to deactivate the ligaments in order to simulate the surgery itself. In the third step, what we can see here, it is really quick. Instrumentation as well as the body weight loads are applied in order to simulate a stand-up process of a human. And finally, there is a steady state body weight that it is loaded in order to see how the model behaves after a while. And now we will focus in the validation itself and the FEM results. First of all, we can see here that the two different configurations that my teammate Anna previously commented, one of a standard deviation of 0.7 and another one with 1.4 in absolute value. Each of them leads to a different curvature architecture. And the results, first of all, we need to consider the lumbar lordosis angle and also to verify it. We can see, for example, at the left of our screens that the lumbar lordosis of 49 degrees leads to a gap score of 3, and the one of 69 degrees leads to a gap score of 7. Gap means indeed the global alignment and proportion, and what it does is to predict the risk of mechanical complications. And we can see that in the 69 degrees, it is bigger than in the one of 49. So the risk of these complications will be bigger. After this, we also need to make the comparison between the stresses that the rods suffer. For example, here they are shown the results for the titanium rods in the 49 degrees and the titanium rods in the 69 degrees. Here we can see that it is 2.5 times greater in this second configuration than in the first one. So the rod breakage risk is higher as well. We also need to make a similar assessment based on the maximum principal stress based on this kind of angles in the annulus fibrosus, specifically in the vertebrae compressed between the L3 and L4. And we can see as well that the maximum stress is suffered in the configuration of the 69 degrees. So collecting all these conclusions, we can say that these 69 degrees are far riskier than the one of the 49 one. Finally, in the vertebra itself, we can see similar behavior, since we can appreciate here some focal points corresponding to intermediate values of stresses, while in the 49 degrees, we only have blue colors corresponding to really low stresses. And finally, we need to bear in mind also some implications when the surgeons overestimate the lumbar lordosis. And for example, here, one option would be to employ different rods based on not only in titanium, but also in chromium cobaltum. And what it will do is to decrease the risk of failure, but it would be a trade-off, since the risk of proximal junction failure would increase. And the implications also are suffered in terms of spine mobility, because we would have less mobility and less stress on the annulus fibrosus, as well as in the vertebrae, as we can see here. The stress gets reduced, because now the 69 degrees employing this kind of material is less than the titanium original one. And finally, we would also like to consider two final facts. The first fact is the role of patient's weight, because it is an important factor in order to predict the root breakage. The higher the patient's weight, the higher the stress in the T-rod. As we can see here, it increases from 931 till 976. And in terms also of AF fibers, it increases as well, because as we can see here, there is a slight increase, a focal point corresponding to dark orange, almost red, and here we haven't. So we can conclude that if the patient weight increases, the stresses behave concomitantly the same fashion. And in terms of vertebrae, the behavior is similar, because here we are appreciating some new focal points corresponding to an increase in the maximum stress. And what we can do, what we can conclude, thanks to all these slides, is that patient weight is an important factor to predict vertebral stresses. And finally, the last slide of results is the lumbar flexion of 20 degrees. It has some implications, because if we make this extra flexion, what we are doing is to obtain a full line in the annulus fibrosus structure of maximal stresses. So what we can conclude is that the statistical shape modeling created two lumbar models, one for simulations and one for kinematic constraints. In the first surgery, that is corresponding to the 49 degrees, there is no need to correct the orientation, because the surgeons already think that this kind of degrees is nice enough. But in the second surgery, that corresponds to the 69 degrees, the surgeons correct indeed the spine curvature, because this kind of configuration is not a safe region. It will lead to higher stresses as well as a higher gap.
Now I will come to the conclusion, discussion, and the future work. So our main conclusions are that the weight of the patient had a significant impact on the rod and stresses, that the chrome cobalt rod was much stiffer than the titanium rod, so then there were less displacements and also lower the stresses, and that the spine morphology, thus the lumbar lordosis angle, had a huge impact on the stress distribution also in the screws and rods. So now we are wondering what happens above the L1 when we use the stiffer rod. Might the proximal vertebrae suffer? And also, are there other angles that would fit better, maybe 53? So then the future will be for us that the FEM workflow can support the gap factor as it considers the material properties. The mesh morphing is very useful for subject-specific implant design, making that efficient. Validation is still necessary, and the pipeline as it is can be applied to implant design. In the end, I would like to thank all of you for listening, and also thank Morteza for the pretty cool hands-on course last week, and also thank the rest of our group who helped us get all this together.
Thank you very much as well.
Fantastic, congratulations on all the work done on the presentation. So let's move then to the last hands-on 7 on medical device modeling.
Okay, can you hear me?
Yeah.
Okay, yeah, I'm sorry because I don't have camera in this computer, so you won't be able to see me, but if you hear me well, that's enough. Okay, so I'll start with the presentation for the hands-on 7. During this week, we've learned how to use the open source MATBAL toolbox GIBBON, which is a very versatile toolbox that allows you to do plenty of tasks such as surface meshing, volumetric meshing, finite element analysis, for which uses the finite element package software VPHi, which is also an open source, so it's full open source, everything. Or as you can do another thing such as image segmentation or so on. So it has been developed by Kevin Moorman, and well, this is the list of participants that have attended to this hands-on. So during this week, we've been working separately in 3 different groups, and each group has studied a different pathology. And well, we have been focused on the implementation or how to use the code instead of the physics of the problem. So if you can pass to the next slide, please. In particular, our group has been focused on the study of the pathology of a diabetic foot, which is the development of plantar foot ulcers that occur due to a loss of sensitivity at the foot at some diabetic patients, and also because of a loss of blood circulation that implies a worse healing process in this kind of patients. One usual solution for this pathology is the development of a foot insole, that redistributes the stresses and the contact at the foot sole in order to decrease the contact at the areas where the bone is closer to the skin, that would be the frontal part of the foot and the heel area, and to increase the contact in the center part of the foot, which has more soft tissue. So this is a problem that typically is solved manually, but with GIBBON, we've been able to automate this process, and starting from real medical images to reconstruct the geometry and obtain an automatized patient-specific geometry for this foot insole. What we have been doing during this week is to test different scenarios for this insole designing process. At GIBBON, there were already implemented 3 different cases, what are the first 2 images that you see at the bottom part, and the left and the center part, which they created an homogeneous thickness insole, and they studied different stiffness variation. There were 2 cases where the stiffness were homogeneous, and it was already also a third case where the stiffness has a special variation, so that the stiffness was higher. So the insole was stiffer at the central part of the foot, where there is much more soft tissue, and it's softer at the parts where the bone is closer. And what we saw is that the results for these 3 cases were pretty similar, even for the spatial variation stiffness case. And what we try to do is to improve this geometrical design by implementing, as well as the spatial variation of the stiffness, another spatial variation for the thickness. So now, apart from being stiffer at the center part, it's also thicker. And apart from being softer, the bones part are less thick, so that we can create an extra gap at these areas so that the contact is minimum. And what we've seen is that we can modify completely the stress distribution. Well, here it is plotted the strain energy density function, which is very similar to stresses. So with this approach, we can completely modify this distribution and get more contact at the central part of the foot. That is what we were looking for. So it seems that the geometrical aspects in this designing process in particular has a greater impact than the material properties of the insole. And well, that's basically what we have done. We wanted to, before we continue with the next group, we also wanted to outline once again that we have found very interesting the great versatility that this toolbox has, because it is very easy to modify these patient-specific designing processes, and it has a very low computational cost, and we have found it really, really interesting. So now I suppose another colleague will continue with the next group work.
So the overall idea of this subproject was to utilize coded modeling to create an automated and optimized process to produce a patient-specific implant. Without the automation, this would be a very laborious process, as lots of steps would have to be done manually. So the pipeline takes patient CT data and creates an implant from their geometry. And so first we segmented the geometry using the CT data, and we created a plane which cuts off the femoral head. If you could go to the next slide, that'd be great. Thanks. The implant is then produced based on the geometry of the patient and inserted into the femur. Meshing is done, and at this stage in the pipeline, the user can specify the type. The material properties are taken from the grayscale of the CT scan and applied to the model. The boundary conditions can then be applied, and the model is ready to be run. The specific parts we added here were deriving the geometry from the CT data and including subject-specific mechanical properties. We then also added some optimization to the pipeline. So the parameters of the implant can be optimized. As an example, we focused on the stiffness of the implant. So to do this, we used a mechanoregulation algorithm, which predicts the bone remodeling over time. We applied a force to the femoral head and outputted the strain energy density. And then this was related back to the model and run iteratively. So the remodeling that takes place is minimized. And the idea here is that you could change the implant design, for example, changing the shape, and then run it again to see which implant produces the least remodeling. From here, we could take advantage of additive manufacturing to 3D print the implant. So basically, the idealized pipeline goes from having the CT data to the physical patient-specific implant. Unfortunately, we did a small miscalculation on how long it takes to run, and it is actually still running at the moment. So we don't have any physical results to show you, but instead just the steps we took to get there. There are a few things that are left to do in this pipeline. For example, we know the shape of the implant is not optimal, but what we have created is like a baseline from which the optimization can go from. So overall, the project was really fun and we learned lots. However, could you go to the next slide? It definitely did not always go to plan. Thanks for listening, and I will pass you on to the next sub-project.
That's great, thanks. So our project was around the FSI modeling of aortic coarctation. So coarctation is just a congenital heart disease characterized by the narrowing of the aorta in the patient, as you can see in the bottom left image. And this narrowing increases the resistance to flow in the aorta, which has several implications. There's an elevated velocity jet, as we can see in this case, up to 3.5 meters per second. And the extra resistance causes elevated pressure in the upper part of the aorta and restricts perfusion or blood flow to the lower limb of the aorta. Any repair for this, which may be a stenting depending on the age, or a balloon expansion at the stent, basically seeks to alleviate the constriction, but comes with long-term morbidities and the risk of persistent hypertension, and even a risk of re-stenosis, so the constriction coming back. So the question is, how good is good enough for the expansion, and what will the impact of a given repair procedure be on the flow and also the risk of morbidity later in life. So this is where the in silico modeling came in, using the GIBBON toolbox. And in this project, Benjamin and myself seek to automate a pipeline for the development or for the setting up and running of a fluid-solid interaction simulation between the fluid in the aorta and its wall, with the intention that this pipeline could be used for automated bulk processing of patients and parametric studies of repairs. And the point being that the user input is really minimized through this process, and output is done so in a timely manner. So I'll hand over to Ben just to discuss how our process.
So I think we need to move over to the next slide, please. So we were able to, in an automated fashion, take a geometry and generate a hexahedral mesh using GIBBON. And from that mesh, again, using GIBBON, we were able to write an input script or input file for FEBio. As it stands at the moment, we were able to run simplified elastic simulations on the mesh. We had issues getting the FSI solver to work, but these things are tricky, and to get it done in such a short space of time is quite a task. And because it's now set up in an automated fashion, running simulations on different geometries can be done instantaneously, and it can be used to investigate different surgical solutions and help with pre-surgical planning. So next slide.
Yeah, so thanks so much. So what we hope that you'd be able to appreciate from the 3 studies shown today is really how easy it is to set up a diverse range of simulations, from studies of the foot to the hip and to the heart, and how this can be used for the in silico testing of devices and other parametric studies. So one of the strengths that was common throughout each example is how these automated pipelines can be used for iterative and optimization procedures, and that it makes it possible to run in silico trials. And importantly, by the nature of the setup, the study will be repeatable, rigorous, and shareable. And the best thing is that it's a free and open source. Source pipeline, and ready to be used and contributed to by the community. So we'd like to thank Kevin for his tutoring and support through each of these projects. Quite a task to juggle so many groups at once, and thank you to the organizers for the week. It's been really incredible.
Okay, thank you very much, guys. I think that the work done in all the hands-ons has been very impressive, and the presentations were very nice too. We're a bit late, but yeah, this could have been expected. I will then ask then the 3 members of the jury then to move to the jury room. So Oscar, Tony, and Jose Manuel, are you able to move there?
Not really.
No, so I'll try
No.
to. I will assign you. So not super handy. Okay, I can put you in the waiting room, but it will not help. So Tony is already there.
When I click to the breakout rooms, I don't see anything. Huh?
There is on the bottom, there is jury room. And if you click on the right side of jury room, there is join. Can you see
I don't
it?
have a jury room. I don't have anything.
Me neither. I saw another room, which was...
No, you were there, Tonya. You were in the jury room.
I was the jury room. That one was the jury room. Okay.
Yeah.
I thought it was the participant room because, okay. So I go.
The jury room.
I will go back to that one then.
Okay. Jose Manuel is there already.
Okay. So when I click on the breakout rooms, it doesn't appear anything as jury or whatever. Huh?
Strange. And I assign you two. So I move you there.
Yes.
Okay, great. So in the meantime, and
do we stay here and do we ask? Can people ask questions about the different sessions?
We're a bit late on the schedule. That's the thing. If you want, you can ask questions through the chat. But what we need to do now is actually then to say who won the awards. Otherwise, we will not have time to do it. So I share the screen. Hope you can see it. So as you know, can you see my screen?
Yes.
Okay, perfect. So as you know, we had then the competition for the VPHi Best Poster Award. So we had 10 posters, we had 6 reviewers, and all posters were assessed. Pardon. All posters were assessed against the originality of the work, the quality of the methodology, possible impact of the result, and the clarity. And so we have one clear winner, and the winner is Wabi El-Bouri. And for the poster entitled Multiscale Modeling of the Impact of Clot Microfragments on the porous representation of a full brain perfusion. So Wabi, congratulations. I don't know if you're here.
Yeah, I'm here.
Okay, if you want to switch on your camera, maybe.
You see me?
If you want to switch on your camera.
It should be on.
Okay. Yeah, that's Zoom stuff. I don't see you. Whatever. So congratulations, Wabi. So the award is a prize money of 350 euros and a certificate. So please get back to Aneta, give her all your contact information. And so we'll manage with the VPH Institute for the prize money, and we will send you the hard copy of the signed certificate.
Awesome, thank you very much. That's brilliant.
Yeah. So I think everyone can at least virtually clap hands for Wabi, who has done a great job with the poster. Congratulations, Wabi.
Thank you, thank you.
And so now we are then moving to the next award, which is the Best Hands-On Award. So as you know, we had 8 hands-ons. We had 3 reviewers who are right now deliberating, and they have assessed all the hands-ons against the presentation. according to the following criteria: challenge and multidisciplinary aspects, conceptual explanations, results and critical analysis of the results, and quality of the presentation. Of course, the nature of the hands-on cannot influence this, this stuff. So everything is based on how good you have sold actually your work and the field of work during your presentation. So I think it's really based. I think technically speaking, everyone has done a great job in every team in 15 hours. It's just impressive to see then the level of technological achievement that you have reached, even if you were supported by specialists, of course. That's quite impressive. But yeah, I think that the presentation and the way you defended it, so now will make the difference. So meanwhile they are deliberating, I can tell you what will be the hands-on. So the hands-on is actually a hard copy of the book from Eric Topol, The Patient Will See You Know. I don't know if you know Eric Topol. He's a cardiologist in the US, and he is actually let's say a pioneer person and an expert in coupling modeling technologies with medicine. He's a great influencer in that sense, and he has written different books about the future of medicine and the role of numerical simulations and models. And in this book, basically, so he gives then a new perspective of how tomorrow's medicine and some would say already today's medicine, so is evolving. So thanks to the in silico methodologies. So I hope you will enjoy then this book. For the group who will win the hands-on, I will ask you then to write Aneta, and you provide her with your contact, okay, and with your address, and then we will manage the delivery of the book to the address that you will have provided. So you will have the book and a certificate. Okay. So I will stop sharing my screen. So, well, no, I will let it, and I will go see what happens now in the room. So I'm going quickly in the room to see what's happening there, and I come back. Hi everyone, thanks for your patience. It has been difficult. So now I will give the word to Jose Manuel.
Hello guys, good afternoon. Well done, all of the teams. The works you have showed are all impressive, but we have to select the winner, and the winner is the group 4B, System Biology Atherosclerosis.
So congratulations, guys.
Congratulations.
Congratulations, very well done. So again, send your contact detail to Aneta, and we'll make the book. And the certificate reach you. Okay.
And
so
again, congratulations to everyone. It has been amazing the work you've done, and really, there were several groups very well evaluated, and in the end, the scores were like 1 point below, 1 point above. I mean, no really large differences at all. Congratulations to all of you, and we hope that you have learned a lot.
And we also would like to seize the opportunity to heartfully thank and congratulate all the instructors of the hands-on, because yeah, you guys have done a fantastic job, as it was reflected and through the presentations and the explanations of the attendees. I don't want to say students because there might be postdocs and the groups, but so no, it's there is the roundtable. We're actually late. Aneta is already there explaining people that they should be a little bit patient. So I think you all have the link for the roundtable. So we will close this meeting, and we connect then to the other link. Okay. Bye-bye.
Bye.
Bye. Thank you, Oscar and Jose Manuel for the evaluations.
Bye. Thank you, Jerome, for everything. Bye.
Thank you. Bye.
Bye-bye. Bye. See you at the roundtable.